Blueprint of Life video 20. In this video, we're going to be discussing some reproductive technologies and how they can alter the genetic composition of a population. This video will be a little bit long, and there'll be lots of notes that you'll probably need to take, and then also a few really important questions and examples that we'll have to answer in class. So reproductive technology is any use of technology that can assist or improve our reproduction rates. There's such things, so we can talk about selective breeding, where there's human manipulation of living organisms to pass on what we call favourable characteristics, and that's a deliberate process that we conduct. And then there's crossbreeding, where we select different varieties, resulting in offsprings with hybrid vigour. So some modern technologies that we're going to be discussing in this video include artificial insemination, in vitro fertilisation, otherwise known as IVF, embryo transfer, and cloning. And all of these four technologies lead to a more specific selection of the genetics in which is being bred, and this has been due to more advanced technologies, including genetic engineering. So artificial pollination is the process by which the stamens of a flower are removed and pollen is dusted onto the stigma. Now, when this pollen is dusted, it can be onto the same plant, whether it's just self-pollination, or a different plant, which is cross-pollination. And this pollination was actually used by Mendel in his study of genetics. This allows the breeder to have greater control over the process and therefore the variation or the lack of variation which they want to see. We then have selective breeding, which we see in animals. Then it's the process by which a male is mated with a female, both of which have desirable characteristics that hopefully is inherited by the offspring. In terms of disadvantages, so the advantage is that you're receiving the characteristics in your offspring in which you have selected, but some disadvantages include, along with the characteristics which you want to inherit, there may be some undesirable side effects from each of the organisms which are also inherited. It costs both time and money for this process to occur, not only to artificially inseminate, but also by transporting organisms. And with this, the selective breeding, so allowing the animals to mate, can sometimes cause risk to the animals. So I did say in the last slide that artificial insemination is expensive, but also the process of using someone else's organism to breed with your own is also expensive. But artificial insemination, on the other hand, to selective breeding, is where the sperm from a male is artificially induced into females. And it doesn't have to be just one female, it can be introduced into more than one. And this has been possible because of effective transport and storage processes that have been developed over time. And that includes being able to freeze sperm. So an advantage is that frozen sperm, to transport it, is cost effective as opposed to transporting your organism which you want to breed. And it reduces the danger to animals that might occur during mating, so that they don't actually go through the physical process in artificial insemination. And frozen sperm can be stored for a long period of time. And this allows it to be, or the genetics of that male, to be inherited in offspring in 5, 10, 15 years, which is also important when we look at breeding programs and trying to increase the number of the population. And the advantage is also one male sperm can be used to inseminate into many females. And disadvantages is that there can be many offspring arising from the one father, so the one sperm sample, which can lead to reduced genetic variability, and we know that may cause difficulty if there's changes in the environment. Next is IVF, where egg is fertilized by a sperm outside the mother's body in an artificial environment. So once the zygotes have progressed to an early stage of development, so that's the sperm and the egg outside of the body, then they can be transferred into the biological mother or a surrogate, or can be frozen for transplantation later. If you look at alterations that occur within the genetic composition of the population, there is an increase in the gene frequency of the selected traits, so organisms that have the desired phenotypes, because of the selective breeding. Usually there's an increase in the presence of those alleles because that product is useful to humans. So rather than having lots of alleles that allow the organism to increase its fitness in terms of the environment or reproduction, they're now more useful to humans, so can be used for human purposes. And also the good thing is that fertile offsprings are always produced, which means that these new allele combinations can be passed on to future generations. Some disadvantages, I guess, is that in processes like IVF, where usually it's occurring because of some infertility that is in either the male or the female, this infertility is passed on and inherited by the offspring. Because humans are manipulating the genetics, we can't always determine the exact combination of genes that the organism is inheriting. So if we look at when we selectively breed and we pick favourable traits that we want our offspring to have, sometimes these favourable traits are linked with some unfavourable ones, so therefore they are also inherited. There's a short-term increase in diversity due to this genetic manipulation, but then over long periods of time, there's a decrease as that gene frequency is the majority of the population. And then we have sperm banks, which allows people to alter the genetic composition of a population as one male, as we learnt before, can fertilize more than one female. And people can actually choose the sperm donor they would prefer, which sometimes which leads into the idea of designer babies, and that can see a decrease in unfavourable characteristics, which might also eliminate important alleles. Here we have a bit of information on the process of cloning, and that's making an individual identical to the one that already exists. So we have reproductive cloning, where we're creating a fully developed organism from another mature organism, and they're genetically identical. So in fact, we are creating twins. There's therapeutic cloning, where we use cells that are healthy from an individual, and we produce more cells from that, and especially when they're cloned from early embryos. They're usually used to repair tissues that have been damaged. And then we have gene cloning, where we're producing identical copies of a gene at a cellular level. If here we have again another discussion on the genetic composition of the population. If there are many clones being produced, then we're reducing variability in the populations because these organisms have identical DNA. And in fact reduces the unknown element because characteristics being bred can be precisely controlled. But if there are any introduction of foreign pathogens or disease, illness, any sudden environmental changes, this population is less likely to survive if they're all from the one clone. So the example that we're going to be looking at in our secondhand investigation is in fact the example of Dolly the sheep, in which a cell was extracted from Dolly. It was then it was taken out. The nucleus of the cell was removed from the egg. Then we see the memory cell inserted inside the egg. Then we follow it to an electric shock, which we saw cell division. The embryo is developed and implanted into the surrogate mother. And then we have a genetically identical lamb being produced from the sheep from the mammary gland that was extracted. So make sure you're in class to complete the secondhand investigations, but make sure you also tune in for video 21.